Some people look older than their age, not just in appearance, but in stamina, recovery, and resilience. They feel it. Slower healing, reduced energy, subtle cognitive fog, longer recovery from stress. And yet their lab results come back in normal. Cholesterol within range, glucose acceptable. Liver enzymes fine, blood pressure controlled. So what explains the disconnect? The answer lies in a distinction most people never hear about: chronological age versus biological age. Chronological age counts years. Biological age reflects how your cells are functioning. And cellular aging does not always show up in routine blood panels. One major driver of accelerated aging is cumulative cellular stress, particularly oxidative stress and chronic low-grade inflammation. Over time, this affects structures inside the cell, including mitochondria, the energy-producing centers that power nearly every tissue. When mitochondria become less efficient, cells generate less energy and more reactive byproducts. The result is subtle but progressive: reduced repair, reduced adaptability, reduced resilience. Scientists often track biological aging using markers like telomere shortening, a process where protective DNA caps gradually shrink with stress and replication. But telomeres are only part of the picture, because aging acceleration often begins in systems that standard lab tests barely measure. And one of the most important of those systems is the one that regulates inflammation. Not dramatic inflammation, not swelling or fever, but the quiet kind that accumulates silently over years. Because some people age faster not due to disease, but due to chronic cellular signaling that never fully turns off. Not all inflammation is obvious. You won't necessarily see redness, you won't feel pain, you may not even notice fatigue at first. But inside the body, persistent low-level inflammatory signaling can quietly accelerate aging. This state is sometimes described by researchers as inflammaging, a chronic, subtle immune activation that increases with age and lifestyle stress. Unlike acute inflammation, which helps you heal, this form lingers. It affects blood vessels. It disrupts insulin signaling. It interferes with cellular repair pathways. Over time, it increases oxidative stress, damaging proteins, lipids, and DNA. Routine lab panels rarely measure this directly. Standard cholesterol or glucose levels can appear stable while inflammatory markers remain mildly elevated in the background. One biomarker often associated with this process is C-reactive protein, CRP. Even slight elevations, still considered a normal, can reflect higher long-term cardiovascular and metabolic risk. A longevity researcher once explained it this way: aging is not just about time, it's about the immune system's tone. What raises that tone? Poor sleep, chronic stress, visceral fat, sedentary behavior, ultra-processed diets. Individually manageable, together cumulative. But inflammation is not the only reason some people age faster, because even if inflammation is controlled, cells still depend on something else to stay youthful: energy production. And when the energy factories inside cells begin to falter, aging can accelerate, even when blood tests look perfectly acceptable. Because the next driver of accelerated aging begins at the mitochondrial level. Every cell in your body depends on mitochondria. These microscopic structures convert nutrients and oxygen into ATP, the energy currency that powers repair, detoxification, muscle contraction, and brain function. When mitochondria function efficiently, tissues stay resilient. When they decline, aging accelerates. What's important is this: mitochondrial dysfunction does not show up clearly on routine blood panels. You can have normal glucose, normal cholesterol, normal liver enzymes, and still experience reduced mitochondrial efficiency. Over time, mitochondrial stress increases the production of reactive oxygen species, unstable molecules that damage proteins and DNA if not neutralized. This contributes to a biological process known as oxidative stress. As oxidative stress accumulates, cells repair more slowly. Energy output decreases. Muscles recover less efficiently. Cognitive sharpness subtly fades. A metabolic researcher once summarized it carefully: aging is often an energy problem before it becomes a disease problem. What drives mitochondrial decline? Chronic stress hormones, sleep deprivation, nutrient deficiencies, environmental toxin exposure, physical inactivity. Over years, these pressures reduce mitochondrial adaptability. But energy production is only one layer. There is another process, one that controls how well your body cleans up damaged cells and proteins. And when that cleanup system slows, aging accelerates even if energy levels seem acceptable. Because some people age faster not from damage alone, but from reduced cellular maintenance. Your body is constantly repairing itself. Damaged proteins are broken down. Old cellular components are recycled. Faulty mitochondria are removed and replaced. One of the key processes responsible for this internal housekeeping is autophagy, literally meaning a self-eating. Autophagy allows cells to dismantle and recycle worn-out parts, preventing accumulation of dysfunctional material. When the system works efficiently, tissues stay cleaner, more adaptable, and more resilient. But autophagy declines with age, and it declines faster under certain conditions. Constant nutrient excess, chronic insulin elevation, persistent stress signaling, lack of metabolic flexibility. When cellular cleanup slows, damaged proteins accumulate. Mitochondria that should be removed remain active but inefficient. Inflammatory signaling increases. None of this immediately changes routine lab results. But at a tissue level, the difference is measurable. Slower repair. Less elasticity in blood vessels, reduced muscle responsiveness, greater metabolic fragility. A longevity scientist once described it carefully. Aging accelerates when damage outpaces repair. And that balance, damage versus repair, is influenced daily by behavior. But there is another factor that often surprises people. Two individuals can have similar inflammation levels, similar mitochondrial output, similar lab results, yet age at different rates, because genetics load the gun, but lifestyle pulls the trigger. And the final piece of accelerated aging lies in how genes are expressed, not just inherited. Because some people age faster due to shifts in gene regulation. Long before disease appears, you inherit your DNA. But you do not inherit how it is expressed. Gene expression is influenced by lifestyle, stress exposure, sleep patterns, nutrition, environmental toxins, and even psychological state. This layer of regulation is known as epigenetics. Epigenetics determines which genes are switched on and which remain quiet. Over time, chronic stress, poor sleep, metabolic strain, and inflammation can alter these switches. Certain repair genes may become less active. Inflammatory pathways may become more easily triggered. Scientists now use measures such as the epigenetic clock to estimate biological age based on DNA methylation patterns. In some individuals, this biological clock runs faster than chronological age, and here is the unsettling part. Standard lab tests do not detect this acceleration. You can have acceptable cholesterol, normal fasting glucose, stable liver enzymes, yet cellular programming may already reflect cumulative stress. A molecular biologist once summarized it clearly. Aging is not just the passage of time, it is the accumulation of signals. Those signals come from sleep quality, stress hormones, dietary patterns, movement habits, inflammatory load, environmental exposure. Over years, they influence how genes guide repair, detoxification, and resilience. Which brings the entire question into focus. Some people age faster not because their labs are abnormal, but because their cellular systems are under quiet, persistent strain. The reassuring truth is this: biological age is dynamic. Inflammation can decrease. Mitochondria can improve. Autophagy can reactivate. Gene expression can shift toward repair. The body remains remarkably adaptable. And once you understand that aging is influenced more by cumulative signals than by isolated lab numbers, the more important question becomes, what signals is your body receiving every day? And what are they teaching your cells about how fast to age? Modern lab panels are designed to detect disease risk, not aging velocity. They tell you when something is clearly wrong. They do not always tell you when something is slowly drifting. Reference ranges are built around population averages, and population averages include millions of people who are sedentary, sleep-deprived, stressed, and metabolically strained. So, a normal often means not yet diagnosable, not necessarily optimal. Biological aging accelerates in the gap between optimal and diseased, subtle insulin resistance. Low-grade inflammation, reduced mitochondrial output, blunted cellular cleanup, epigenetic shifts towards stress response dominance. None of these may trigger red flags individually, but together they compound. A preventive medicine specialist once said, aging is cumulative adaptation to stress. That adaptation can move in two directions, toward resilience or toward fragility. The difference is rarely dramatic in the short term. It's directional, and direction is shaped daily. This is why two people with identical lab reports can age differently over a decade. One accumulates repair signals. The other accumulates stress signals. The body records both, and it expresses them over time. So the real takeaway isn't fear. It's awareness, because aging faster is rarely random. It's usually the result of quiet biological pressure, repeated consistently. And once you recognize that, the more useful question becomes, not are my labs normal, but is my lifestyle signaling repair or survival? That difference determines how fast the clock truly moves.